Hi, so welcome to the Marine Vertebrate Genomics session of the Biodiversity Genomics Conference. So if you've been watching the earlier sessions in the Australia time zone, then you'll see there's already been a good representation of some marine vertebrates, but there's lots of them, so we have plenty more to say. So it gives me great pleasure to introduce three presenters for this session. So first off, we're going to have Regine Tionghui Yi from the Singapore Centre for Environmental Life Sciences Engineering in Yang Technological University, Singapore. She'll be talking about the population genomics of critically endangered hawksbill turtles in Singapore and how they unveil a genetic vulnerability and urgent conservation needs. We'll go straight into the second talk, which is Isabel Nakayama from the Institute of Oceanography at the University of Sao Paulo. She's talking about seascape genomics of the shortfin mako shark in the Atlantic and Indian Oceans. And then lastly, we have Roberto Biello from the University of Ferrara in Rome, talking about seahorses in the Anthropocene, conservation gaps and trends. We're going to run the talks back to back in this session and then have a Q&A session at the end. I strongly encourage you, please, to go to the Slido for this session and post in any questions. Feel free to post questions during the talks. If they end up getting answered as the talk goes along, we just won't ask them at the end. You can also upvote any that particularly interest you that other people have asked. So without any further ado, I'll hand over now to the actual presentations.
Hi, my name is Regine and I'm a PhD student from NTU Singapore. My talk is about the population genomics of critically endangered hawksbill turtles and how our study has showed the genetic vulnerability of this population and the importance of conserving this species in our region. Here in Singapore, we are home to hawksbill turtles and we see them nesting and foraging despite being a very small and urbanized island with high shipping activity. They are also the only marine turtle species that nest on our narrow and reclaimed beaches, as we also see green turtles foraging around our coast, but they do not come up to our beaches to nest. We began studying the hawksbill turtles in 2019, and we see about 10 to 20 nests per year. However, out of the 7 global species of marine turtles, the hawksbill turtle is critically endangered. Aside from the many threats faced by most turtles, like the loss of nesting habitat, bycatch, and climate change, the hawksbill turtle is also threatened by the illegal wildlife trade for their beautifully intricate shell, which are often used to make ornamental products, leaving them to be at such desired state and at high priority of conservation. But why does the hawksbill turtle matter in the larger scheme of things? This is because they are actually important to our reef ecosystems by keeping the sponge population in check due to their unique diet for sponges. Sponges compete with corals for space, and thus the hawksbill turtle plays an important role in maintaining their population, allowing the reef ecosystem to thrive, which is also so crucial for protecting the marine biodiversity that heavily relies on our reefs. With hawksbill turtles listed as critically endangered, I hope that my genetic work will help to contribute to the conservation of this species. And so this is an overview of my talk today. I have 3 sections. Firstly, I'll cover a study on the genetic diversity of hawksbill turtles in Singapore using DNA haplotypes from the mitochondrial control region. One unique trait of the marine turtles is that they have geomagnetic imprinting, whereby when turtle hatchlings leave the nesting beach for the ocean, they can also remember and return back to the same nesting beach as adults. And this is also known as natal homing. This allows for the use of mitochondrial haplotypes to be effective since the mitochondria is only inherited through the maternal lineage, and it will also provide a reliable view of the genetic diversity of the hawksbill turtle population nesting in Singapore. Hawksbill turtles mainly nest on the eastern and southern beaches in Singapore, and based on our results from 5 years of sampling, we found 6 haplotypes from a total of 39 nests and from 5 nesting sites and 7 foraging individuals in Singapore, revealing a small genetic diversity in the hawksbill turtle population. 5 out of the 6 haplotypes were found to be present in the Java Sea, in Indonesia, and one of the 6 haplotypes were found to occur in Malaysia. These 6 haplotypes are also closely related to one another, with only 1 to 3 nucleotide differences among them. And when compared to the database found in the Indo-Pacific region, they all belong to the same clade IP3. The overlap of mitochondrial haplotypes in the Singapore's hawksbill population in those in the Java nesting sites implies that these populations could be maintained under the same management unit and emphasizes the importance of international collaboration for marine turtle conservation efforts. But of course, since we're in a genomics conference, we can't not talk about genomes. And so in my next section, I will share about our development of the de novo genome assembly of the hawksbill turtle. As we know, the mitochondrial DNA has its limitations, like not being able to identify individuals or really map out the individual relationships with one another, as well as not being able to understand the population structure and genetic contribution from both the males and the females in the population. We can also see how much genetic information we are missing out when we are only using the 800 base pair mitochondrial haplotype in comparison to the whole genome of the hawksbill turtle, which is about 2.2 billion base pairs. During this time of our project, there was also no published hawksbill turtle genome yet, and so we have decided to produce one of our own. Here is the workflow of producing the de novo genome assembly, where we sequence a hawksbill turtle DNA of high quality using both long-read ProMethION as well as short-read Illumina HiSeq-X sequencing. The ProMethION long reads were base called using Bonito and then assembled using Shasta. The short reads were then mapped onto assembled long reads for error correction by polishing. And the resulting hawksbill turtle genome assembly can then be used as a reference genome for the population data using whole genomes, which I will cover in the last part of my talk. We then compared our genome with other published genomes, and these are the synthenies of our genomes against other published genomes of other marine turtle species. We have the green and leatherback turtle by Bentley et al., and we also have the published genome of the hawksbill turtle by Guo et al. That was sequenced last year. We also see a high degree of synteny across these genomes, suggesting a conserved mode of evolution across marine turtle species. Our genome is resolved at 2,750 contigs, with the largest contig at 224.2 MB and an N50 of 3.4 MB, and a coverage of 57.9x. With our reference genome, we then studied the hawksbill turtle population in Singapore using whole genomes. With a data set of 35 genomes with a high coverage of 30x, we're able to get more than 11 million SNPs, which is a vast difference compared to the 6 variable sites using 115 mitochondrial control region samples. The average number of SNPs for each genome against the reference is also about 4.5 to 5 million SNPs, which is higher than that of the global human population, despite being a critically endangered species. Using our whole genome population dataset, we're also able to understand the relationship between nests using IBS, identity by state, and this analysis revealed a high degree of inbreeding, with more than 1/3 of relationships in the population exhibiting at least third-degree relationship, and that is of first cousins. We also discovered the presence of multiple paternity within the nest, with samples from the same nest exhibiting both full sibling and half-sibling relationships. This was not the only exciting part, because when zooming in closer into this nest of siblings, we also discovered using IBD, identity by descent, that instead of half-siblings, they were reviewed to be more like 3/4 siblings, suggesting that the fathers of this nest was also closely related. Our inference of demographic history using MSMC analysis, which stands for multiple sequentially Markovian coalescence, also revealed a significant decline in the effective population size of about 8-fold reduction occurring approximately from 60,000 years ago to 10,000 years ago, particularly from the marine isotope stage 2 from about 29,000 years ago the population size declined significantly and remained at its lowest, while sea level and sea surface temperature rise rapidly during the last deglaciation period, with the population not recovering until climate conditions stabilized. The population size was slightly suppressed by the rapid sea level rise during the last deglaciation period due to the submerging of nesting sites, and the population only increased when sea levels were stabilized. This result shows that climate change emerges as a significant challenge for hawksbill turtles, and our findings highlight the profound impact of environmental change on the hawksbill turtle population and the vulnerability to ongoing and future global warming. Whole genome screening for positive selection was also conducted using IHS on 18 unrelated individuals. A total of 94 genes were detected under positive selection, and our functional enrichment analysis revealed that these genes are predominantly involved in pathways including cellular transport, cellular signaling, olfactory perception, as well as diseases. Cellular transport and signaling play a crucial role in environmental sensing and response, like maintaining cellular homeostasis in high salinity conditions. Whereas the olfactory perception is important in assisting foraging behavior, specifically to detect airborne and waterborne scents. 4 pathways were also found to be associated to cancer-like diseases, which is also interesting as marine turtles are vulnerable to this tumor disease, also known as febrile papillomastosis. This disease is caused by a herpes virus infection, which results in tumors in the organs and on the skin of marine turtles, which results in death due to its debilitating nature. These findings show how genomics can reveal important insights of a population that will be crucial in directing conservation management efforts. And with that, I would like to ask you all to check out our preprint in bioRxiv, and I've come to the end of my talk. This work would not have been possible without the help of these people. And thank you.
Good morning. My name is Isabelina Caima. I'm from the Marine Genomics Laboratory at the Institute of Oceanography of the University of São Paulo, Brazil. And I'm here to talk to you guys about the seascape genomics of the short-fin mako shark in the Atlantic and Indian Oceans. To begin our conversation, I would like to introduce the short-fin mako shark. It is known as the fastest individual in marine environments and known for its very highly migratory capabilities. It is found in all tropical and temperate oceans, as well as known as being an apex predator of its food webs. It is also fairly studied, having its mitogenome and more recently its genome sequenced. And it's known for its long gestation periods, as well as late sexual maturation, which is very important when we're talking about the conservation and management of a species. That said, it is classified as endangered by the Red List of the IUCN, which is the International Union for Conservation of Nature, and which is a classification a little below extinct. And this varies depending on region, but this is an overall global classification. This happens mainly because of the fishing industry, which can have the industrial fishing and mainly bycatch being a factor of the reduction of the populations of the short-fin mako, but also sports fishing and mainly finning, which is a practice that is economically encouraged due to its high demand in some countries where they eat and consume the shark fin soup, which is a delicacy, making it an economically desirable product. So why should we study sharks? Today, we know that sharks have a high ecological importance, being responsible for maintaining the health of many marine and ocean systems due to their role in their food web. So they control populations which help maintain an ecological equilibrium of their marine systems. So for conservation, we need to first understand how we can study these individuals so we can make adequate management and conservation strategies. So for this species, for the short-fin mako shark, we first need to identify their fish stocks and populations in order to think further of management strategies. So there are some studies already done using mitochondrial DNA in some regions like the Pacific Ocean and the South Atlantic or the North Atlantic, but not in the same area of studies that we're doing in this project. So for this study, we're utilizing SNPs, which are single nucleotide polymorphisms, which is a type of mutation used where we analyze a variation of a single nucleotide of a DNA or RNA read, and they're very well used for identifying populations. This differs from the previous studies mentioned where they use mitochondrial DNA. SNPs are today's more recent way of identifying populations and are the targeted method of this study. In order to identify the SNPs, we first use the DNA sequencing using ddRAD-seq, where we use the digestion of the DNA, the extracted DNA, using two restrictions enzymes, where they attach themselves to the extracted DNA and isolate of DNA fragments, which will be then analyzed later. And we also use RNA-seq, where we took an RNA strand and we converted it to its complementary DNA in order to analyze later as well. So with these extractions, we're going to be able to identify the SNPs and answer the question if the short-fin mako shark in our study areas are in panmixia, which means random fertilization of all individuals in this study area, or if they are divided into populations, so if they have a certain population structuring. To answer this question, we collected 235 tissue samples of the short-fin mako shark. We prepared the ddRAD-seq and the RNA-seq libraries, and we identified and isolated the neutral and outlier SNPs. This was done so we could study the genomic diversity levels and possibly identify population structuring of the individuals in these areas. For the RNA-seq, we had the 235 initial samples where we identified 1,200 SNPs, and after filtering, we remained with 121 individuals and 555 SNPs. And we did 3 methods of outlier identification, and the outlier SNPs in common of all 3 methods were the outlier SNPs that we remained with. And for the RNA-seq, we had 8 outlier SNPs. For the ddRAD-seq, we had the 235 initial samples, but with only 401 SNPs. And after filtering and analysis, we had 136 individuals with a remaining of 216 SNPs. And after outlier identification, we didn't find any outliers that were common in all 3 methods of outlier identification. So for the fixation index, we studied the individuals for the RNA-seq as well. And here we see that the FST generally goes from 0 to 1, but here we have values up to 0.06. So we see that there is very low values of the fixation index, but there is a differentiation between the areas studied. So here we see that there is some grouping happening in between the northern hemisphere region, so this is Cabo Verde, and this is the East Atlantic Ocean, and the southern hemisphere with Brazil, Indian Ocean, South Africa, and South Atlantic Ocean, with the exception of Portugal, which seems a bit of an outlier here, and this might be due to the sampling quantities that we had. And for the ddRAD-seq, we see similarly here that there is low levels for the fixation index, but there is differentiation of the genetic diversity between the sampling areas. And here we have a good grouping as well of the southern region, as well as the northern region, the northern hemisphere region. So we also analyzed the genetic diversity. So we use the observed heterozygosity, and we see that the highest values of heterozygosity is for the individuals in the northern hemisphere in comparison to the southern hemisphere. And we see negative values of the homozygosity, which is the inbreeding coefficient. So this shows us that there's no inbreeding happening, that even though this is a endangered species, so this is a good sign for the for future conservation, for example. For the DD-RAD-seq, we also have values of the northern hemisphere that are showing us the highest values of observed heterozygosity in comparison to the southern hemisphere. And also we have negative values indicating no inbreeding happening. So for population structuring for the RNA-seq, we did an ancestrality test. And here we see that the northern regions have very similar ancestrality in comparison to the southern regions. So they have genetic materials being shared here. And they also, but they're all green and yellow. So we can see that there is gene flow happening, but there is some structuring being represented in terms of ancestry. This is a Bayesian sample, and we also did a multivariate with the principal component analysis. And we have very similar results with the clustering of the southern hemisphere individuals with the northern hemisphere individuals. And here we have a visual representation of what that means. So southern hemisphere and northern hemisphere are being grouped, even though we have very similar colors, representing that there is indeed gene flow. We have very similar results for the DD-RAD-seq. So here we have the northern hemisphere and the southern hemisphere being grouped. However, we do see that there seems to be more gene flow. And for the PCA values, we also have, this is also represented by the clusters being a bit closer together, but there is still clustering of the southern and northern region. And this is a visual representation of the groupings that we see, even though we see more gene flow in the DD-RAD-seq, there's still some structuring being observed. So here's the side-by-side comparison of the RNA-seq and the DD-RAD-seq. And here we can say that since all of our other results show that there is some structuring, we can confidently say that there's structuring of two populations of short-fin mako shark here. So this is very important when we're talking about conservation. So because organizations like the ICAT, for example, can create management strategies that are more targeted and for those populations and can have long-lasting effects in terms of the preservation of the short-fin mako. So for this, we need to we first identified that there are two populations. So now we need to ask ourselves, how come there are two populations for this highly migratory and fast species? So here we tested 21 oceanographic variables. We remained with those with correlation below 0.7, so we don't have a redundancy in our results. And some of the variables that we remained are bathymetry, chlorophyll, sea surface currents, pH, cloud cover, nutrient salinity, and temperature. And besides the oceanographic variables, we also tested the spatial relationships. So the shortest by-water path, this is the visual representation of the shortest by-water path between all individuals collected and sampled. And we also tested for the distant Base-Morn's aim factor maps, the DB-MAPs. So in order for us to unite the genetic diversity that we saw and the geographic and oceanographic variables, we did a redundancy analysis where we tested for RNA-seq. Here we saw that there is clustering of the southern hemisphere here in the first two quadrants, and then the northern hemisphere here below. And we see that there is still, and we see that the distance are main factors for this types of clustering. However, we do see an influence of the chlorophyll here in the separation of these two populations. And for the DD-RAD-seq, we see similar values here with the southern hemisphere being clustered and the northern hemisphere being similarly clustered, with also chlorophyll acting as a factor. So what is the take-home message? Here we see that we can confidently say that there are two populations in the Atlantic and Indian Oceans that have high gene flow and low population structuring, and that they have a moderate gene diversity, which we saw in the observed heterozygosity values. We also saw the chlorophyll indicates to have an influence on the population structuring. However, we do see that there is a greater influence of isolation by distance than isolation by environment. And these are the financing institutions that help make this project viable. This is FAPESP, CAPES, and the FCT, and the partner institutions being a few universities in Brazil and Portugal. And I would like to thank everyone for watching my presentation. Here's my email for references or questions, and our laboratory's Instagram page. Thank you very much.
Hi everyone, I'm going to present today a research on seahorses. Particularly, we are focusing on the impact of the Anthropocene on the species and how we can integrate conservation gaps and trends. As many of you know, seahorses are fascinating marine fishes that have become popular due to their characteristic appearance and the unique biology, including, for example, the male pregnancy. Probably not everyone knows that there are currently around 46 known species of seahorses, all of which belong to the Hippocampus genus. And these species trace their origins back to the Indo-Australian archipelago. And they emerged during the late Oligocene, approximately 25 million years ago. Seahorses are found in a variety of marine environments, from shallow seagrass beds to deep water. However, despite their diversity, these species are currently facing many serious threats. Pollution, such as plastics, is severely damaging their habitats, but also overfishing continues to reduce the population size of these species. And also, finally, the habitat loss with the destruction of marine ecosystems like coral reefs and mangroves are increasing these pressures. And these factors altogether are pushing many seahorse species closer to the edge of extinction. So what we know today is that approximately 35% of seahorse species are at risk of extinction. And this number could increase if current trends continue. Also, despite these species are very popular, it's alarming how little we know about them. Nearly 39% of species of seahorses are classified as data deficient, meaning that there is a limited information or no information to properly assess their conservation status. Moreover, much of the current conservation focus is based on changes in habitat and population size, but one critical factor often is overlooked, and it is the genomic diversity. The genomic diversity represents the basis for evolution, and without considering it, we might be underestimating the true extinction risk of these species. So our research aims to address these gaps by contributing to more effective conservation strategies for these species. We did this by analyzing data on multiple aspects of the biology of the species and the ecology of the species. And we focus on several key questions. First, what is their geographic and phylogenetic distribution? We want to know where species are located, how they are related. Another important question is, what is the scope of the current spatial protection? This can help us to understand the level of protection these species currently receive through spatial protective measures, for example, marine protect areas. Another question is, what is their genetic health and evolutionary potential? To know this is essential for their long-term conservation. And lastly, are these species resilient to current and future challenges? We want to know if seahorses are sufficiently able to face the current and future environmental challenges posed by human activities. So to answer all these questions, we used a comprehensive data set. We downloaded data from these two repositories, GBIF and CBI. And our data set is composed of three main components. First of all, we obtain species occurrences for 35 species, and we also obtain the extent of marine protected areas that overlap with the habitat range of the species. Then for the phylogenetic diversity, we obtain approximately 1,000 ultra-conserved elements in order to estimate the evolutionary diversity across species. And finally, we obtained whole genome data of 358 samples from 21 species, and then we mapped all this data to the Hippocampus erectus reference genome. So by analyzing this multidimensional dataset, we took a transdisciplinary approach. So we integrated data on biogeography, evolutionary history, genetic health, and also the management plans that are currently in place. And this broad perspective, we believe, will allow us to make more informed proposal for conservation. So let's now take a look at some of our findings. And one of the most important key result was the creation of updated distribution maps of the species. And this was made possible in part by citizen science contribution through the use of the platform iSeahorse. These maps allow us to ask important questions about the efficiency of current conservation efforts. And for example, one important question is how much of seahorse habitat is actually protected by marine protected areas. Unfortunately, our analysis show that on average, only about 7% of seahorse habitats are protected, and below 20% in most of species. And this level of protection is clearly insufficient given the scale of the threats that these species are facing. And it's even more concerning is that these protection levels are not really well correlated with the IUCN conservation status, as you can see from the plot. In fact, species identified as being at higher risk of extinction are not necessarily the ones receiving the most protection. So next, we analyze the maps of phylogenetic diversity and endemism based on these ultra-conserved elements. And as we expected, with the Indo-Australian archipelago, showed the highest level of both phylogenetic diversity and endemism, the red areas on the maps, which makes sense considering that, as we saw, that this area is the center of origin of all seahorse species. Another important question is how much of this breadth of evolutionary history is protected by current policies. And our results are still not really encouraging on average because less than 3% of species is protected. And this will decrease even further when we consider regions at the higher phylogenetic diversity or endemism. This region, unfortunately, corresponds to the region with the most affected by overfishing of these species. So our comprehensive analysis of the whole genome data across 21 seahorse species revealed a wide range of genomic diversity in the species. Some species exhibit high level of genome-by-wide diversity, while others show a very low diversity. This variation is understandable given the wide range of species that we considered, and also all the species have a unique adaptation, also different evolutionary background. But however, what is particularly interesting in this analysis is that the genomic diversity does not align clearly with the IUCN conservation assessment. In particular, we found species classified as vulnerable in orange with high genomic diversity, while other species in the same category show particularly low diversity. And similarly, we estimated the proportion of the genome in long runs of homozygosity, and we observed high variability pattern across species. Again, some species show a low level of inbreeding with 10% of the genome in a row, while others show almost 80% of the genome in a row. That means that they have a very high level of inbreeding. And this variability confirms our findings on genomic diversity and tell us that these species are facing different level of genomic erosion, but also indicating that for now, most of your species are not showing strong signs of it. Also here, we don't really see a clear connection between the level of inbreeding and the IUCN assessment. We also measured the genetic load across 21 species. Genetic load refers to the burden of deleterious mutation that can be used as an indicator of genetic health. Interestingly, most species show a relatively low genetic load in their homozygous state, that is the realized load. This may suggest that there was a past purging of deleterious mutation that removed this mutation. However, several species carry high number of such dangerous variants in their heterozygous state. It means that there is high potential of masked load. While this mutation remain masked for now, that could become problematic if exposed through inbreeding in the future. So at the present, we can say that this does not seem that seahorses are at limited risk of genomic erosion. However, we must be aware of what is known as drift depth, which refers to the time lag in evolutionary genetic change. Genomic erosion only becomes evident many years after a population has experienced a decline, and seahorses are facing their main threats only in recent decades, so they could similarly experience this delayed effect. And in summary, we can say our research provides multidimensional insight into the diversity of observation of these species. We show that despite being charismatic animals, their current level of protection is poor, and also identified area and species that should be the priority for conservation. Most of seahorses do not show a strong genomic erosion, but their demographic decline might be too recent, so we have to be aware of the potential drift depth. Additionally, their genetic status is not well described by current conservation indicators, as we see. So genomic erosion seems not to be an immediate concern in this species, but reduced diversity, increased masked genetic load may reduce the viability in the future. So finally, our study provides an example of how open science practice can contribute to proper evidence-based assessment and management of biodiversity. You too can contribute directly to these efforts by sharing your data and observation. So you can help fill the data gaps using the platform iSeahorse. If you have seen now a seahorse while you're doing diving or snorkeling, you can record it, improve our understanding of this incredible species. So finally, this research would not have been possible without the collaboration and contribution of many colleagues who provide data, analysis, and ideas. So I want to conclude here, and I want to thank you for your attention, and I look forward to any questions or discussions you may have.
Okay, so thanks for all our speakers for some very interesting talks there. We have two of them online with us for the Q&A session. I believe it's very early in the morning in Rome, so Roberto might be struggling to join us. I do encourage you to email him if you've got any questions. And likewise, anyone who's watching the recording of this, please do follow up by email if you have any questions or insights for any of our speakers. So with that, I'll crack on with the actual questions. I'll start with Regine as first, and your questions at the top. So first one, thanks for a great talk. I'm curious as to the genes you found that were associated with disease. Do you know the potential mechanism that links them to viral induced tumor susceptibility? Are they viral response genes?
Thanks, Emma, for the question. So we did find the genes. They are TLR8 and ZNFX1 genes. So this is a very recent part of our research, so we haven't really looked into the mechanisms. But for this disease, it's actually very well known in another species, the green turtle, as we see many more occurrences of tumors on the green turtles compared to the hawksbill turtle. So I do believe that perhaps the mechanisms could be very similar, but we haven't looked much into it yet.
Cool, thanks. Okay, next question is one from me. So I was curious, with you got your resequencing data, do you find that there are lots of either unmapped reads for different individuals or bits of the genome where there were no reads mapping? As someone who sort of makes a lot of reference genomes, I'm always interested in how much that single reference is useful for everything, or whether you feel like you need to go to a pan-genome approach.
Yeah, thank you for the question. So I do believe that a lot of, so we did have a few samples with low coverage and also a lot of unmapped reads, but I think it's mostly likely due to the quality of the sample that we sent for sequencing, but most of the other samples map really well. And because as we compared to other species, such as the green and the leatherback turtle, they all had very high synteny and high conservation of the whole genome. So I believe there's no need for a pan-genome at this moment, but I think it will still be interesting to take a look at. Thank you.
Okay, sticking with you, next question. Thanks for presenting your work. You looked into IBD and IBS. Did you also look into recombination breakpoints in the population across the chromosomes for linkage groups?
Thank you, Gibran, for your question. So for us, we didn't look into the recombination breakpoint because for now, our genome is not at a chromosomal level yet. So I think it will be a bit difficult to do that, but we will love to explore that in the future. Thank you.
Great. Okay, we'll switch over to questions to Isabel now. If you have any more questions for either of our speakers, feel free to keep adding them to the Slido. So Isabel, for the population structure analysis, did you try looking at more than 2 population groups? And I was wondering how much of the variation in that PCA plot you showed, how much of the variation did that actually explain?
Okay, so for Seascape Genomics, we tend to use the Bayesian information criterion, which is the BIC or BIC. I'm not sure if that's BIC is the global terminology, but it's the Bayesian information criterion, where we, through that test, we analyze the K value and then analyze some of some tests based on that. So if for example, the data showed that there's a K equals 2. So we use some of that for like some of the tests, but other tests also showed naturally that there's 2 populations. So there was no need to keep on testing more. And if there were more populations, other tests would have shown even without the beak analysis. So that's what we use as a base, which is very common for like Seascape Genomics.
Cool. Okay, following up with another question from Shannon Corrigan on the structuring. Thanks for a great talk. Nice to see this work and the structuring between Northern and Southern hemispheres maintained in genome-wide analyses. I'm curious, is there a chance your Portugal samples were fished in the Southern hemisphere?
Okay, so that's a very good question because we have a very low sample number from the Portugal and Brazilian fishing, but that's one of the problems from the any studies working with sharks, because we kind of depend on partnerships and to make that happen. So we rely on the trust that involves like the researchers from other industries or universities. So we trust that that's actually correct data that we have, and there's no way of knowing because we actually just have to trust that there are partnerships that we're using are actually trustworthy, factile. So, but Portugal has an incredible institution, so there's no way to doubt that they are not, that the researchers are not actually collecting factable material. But the data does show that there might be at least some contamination, which is very common, as most of us probably already know that in genomics, that's what happens. Yeah.
Cool. And one last question, which is again for me. So, were you surprised by the influence of chlorophyll you saw on the population structure? And being ignorant, is there anything to do with like the food and what the food is actually eating that you think might be influencing population structure, or?
Okay, so that's a very good question because when we ran some tests from the RDA specifically, which is the redundancy analyses, we did get some different results from what I presented. So that's a very good thing to point out. We did receive that chlorophyll, when we first test like RDAs, I'm not sure if everyone's familiar with this, but we first test against allele frequencies, and then for both geographical and oceanographic variables. Then after we test for each other, we test them amongst each other. So chlorophyll showed to be statistically significant, but in the RDA, once tested against the geographical variables, they showed not to be significantly statistically significant. So what I presented that was a little bit different from the actual results that I reran this week. So even though they do have an influence in allele frequency, once compared to distance and geographical variables, they lost some of their statistical significance. So we do see that isolation by distance is far more valuable for the short-fin macro shark gene flow than oceanographic variables. So some variables did show to be, some oceanographic variables did show to have a statistical importance for allele frequency, but once compared to the geographical variables, they were not statistically significant. So that's very important to highlight that I recently rechecked.
Great. That's the excitement of having hot off the press data. So I think we've got through all the questions in the Slido now. So again, if you have any other questions you think of after the fact, please do get in touch with our speakers by email. It just leaves me to thank our speakers once again for a really interesting session, and encourage everyone to tune back in about 10 to 15 minutes for the amphibian research and conservation session. So thank you.